I'm the Chief Business Officer, as was indicated, at uniQure, which is a AAV-based gene therapy company. And I will go through our company overview. This is a standard forward-looking statements disclaimer, as we are a publicly traded company, traded on the NASDAQ, although we are based in the Netherlands with headquarters in Lexington, Massachusetts, but we are Dutch incorporated. And our mission, as it states here, is really to deliver curative one-time therapies to transform the lives of patients. Our strategic imperatives as a company, first and foremost, is to develop a robust pipeline that I will review with you today, primarily focused on liver-directed and CNS disorders. And by liver-directed, we mean that we target the liver, although it's not necessarily simply liver diseases. We have and want to maintain leadership in manufacturing. Manufacturing is something that we invested in early on with very significant resources, again, that I'll review with you today. We took a leadership position in insect cell baculovirus-based manufacturing of AAV gene therapy products and have a large facility for those purposes and some very foundational intellectual property to support that. We also have a lot of work going on in enabling technologies, novel promoters, novel capsids, administration technologies, and other things that are very important for the successful delivery of gene therapy, and a strong intellectual property basis for the work that we do. And then, very importantly, we have full commercial rights to nearly all of our programs, with the exception of the programs that we have in partnership with Bristol-Myers Squibb. And we are in phase 3 with our lead program, so commercialization efforts are underway at the company now, early commercial, which I'm responsible for. This is a quick picture of our pipeline, and as I just mentioned, the Hemophilia B Program in the liver-directed diseases is our lead asset in phase 3. And then we have a Hemophilia A program as well, which is in late preclinical development. This program is not a typical factor VIII replacement approach, and I will describe it in more detail. We also have a Fabry disease program in preclinical development that is also, we have multiple, but one of the programs we have is not a typical alpha-gal replacement approach. And then in the CNS area, we have just begun clinical trials with our Huntington's disease program. This is a knockdown approach as opposed to delivering a gene. I'll cover it more in detail, but this delivers a gene that produces a microRNA that then blocks the messenger RNA for the Huntington protein. We also have a SCA3 program as well, also a knockdown based in CNS, as well as other things in that area, and partnered programs with Bristol-Myers Squibb. These are the near-term goals for our company, and Hemophilia B, which is in phase 3. We will be working towards completing the enrollment of the phase 3 program and beginning to generate data there. In Huntington's, we are initiating our phase 1/2 study very shortly. And in Hemophilia A, our plan is to submit an IND in the near future. And then for other programs, we will get IND-enabling tox studies initiated with them. So this summarizes our manufacturing facility. And I think many of you are probably aware at this point that manufacturing is a rather critical capability to have, or at least to have resources that can provide it for you. It becomes quite a significant issue as you go into phase 3. And there's a lot we could talk about here, but what we have done is to create, some time ago, in fact, and have been adding to it, a rather large facility that is GMP capable. We are a company that actually has, in the past, had a product approved in Europe, so we do know what it means to create GMP product for use in people not just in the clinic, but also in a commercial setting. But we have further built this facility out in the United States to support all of the work that we do. It's based on, as I said, insect cell baculoviral manufacturing that's scalable up to this particular facility up to 2 by 2,000 liters, meaning up to 4,000 liters total. And we have very strong intellectual property position in the insect cell baculoviral space. We did a lot of work to really create that field. which has certain very key advantages as compared to other platforms like mammalian cell. There's a great deal more control. It's highly scalable from small scale to large scale without a great deal of changes. And it is also very cost effective and creates very consistent, high quality product when it's done correctly. Another asset of ours, which is very important, is the use of the AAV5 capsid. And the AAV5 capsid has a lot of unique qualities that are extremely advantageous. We have got a lot of clinical experience with it. It's been in 25 patients in clinical settings to date in multiple different indications. It's also being used by BioMarin in their program to treat hemophilia A patients, so a good deal of clinical experience. And what's very important about this particular capsid is its strong immunological profile. It has very low levels of pre-existing antibodies. That is not the case for many other capsids. We are not excluding any patients from our phase 3 study on the basis of neutralizing antibodies. And there's been no T-cell responses noted to date at all. And that is also very important. No loss of effect over time with this particular capsid. So it's been the basis of a lot of our programs, both Huntington's as well as hemophilia. I'll review quickly the Hemophilia B program itself. And our intention here is to create a durable, long-lasting treatment for hemophilia with no peaks and troughs that give patients essentially curative benefits. We are doing this with the Padua form of the factor IX protein. And for those of you who don't know, I can explain in more detail what that's all about. And we have a strong intellectual property position in that particular mutant form. The data that we generated in our small phase 2b study with 3 patients is shown here. And what you see is long, sustained, and steadily increasing levels of factor IX activity, which is the key biomarker for how patients measure efficacy. And these are at levels which are very robust, very consistent, and effectively curative for patients. There's no loss of effect. There's been no need for any replacement therapy whatsoever in these patients, and there's been no bleeding and no requirement at all for immunosuppression, no steroids used prophylactically or in response to anything. So this is a very, very compelling profile for patients. Our phase 3 study is currently recruiting. At least one patient has been treated, and we plan to treat about 50 patients total, and then generate our phase 3 data. Next, I'll cover the Huntington's disease program. This is also a one-time administration. It's stereotactically injected into the brain, and I'll describe further, but it's in the putamen and the caudate. It uses our proprietary uniQure technology to, as I mentioned before, knock down the messenger RNA for the Huntington protein. And we show very good knockdown in the animal models that we've used. And it is, as far as we know, the first such program into the clinic. So we are in the lead with as compared to other programs. What we've shown in animals, this is non-human primates, is very good distribution. So the injection goes into the caudate and the putamen, which is the striatum, the central part of the brain there, and the dark areas show where you see DNA distribution of our microRNA DNA. And it also, however, gets to other areas where it needs to go through retrograde and anterior grade transport and exosomal transport, which is something that we've talked about as a company quite a bit. But it's a very compelling, unique property of our microRNA and of our AAV5 vector used in this way. And it gets to areas where it also needs to be in the cortex as well. And so that's very important. Then we've also done work in a transgenic mini pig, which is not all that mini at all. Some of the details about the pig are on the left-hand side of the graph of the slide. But this is what's transgenic. They've been given human Huntington gene, so we can very, very well measure knockdown. Whereas in the primates, the Huntington gene, there's a one amino acid mismatch with the primate versus the human. So you don't measure knockdown as well. But this is a good model to measure knockdown. And we have done that at both 6 and 12 months in the mini pigs. And you can see in the areas where it is important to see knockdown, we see very robust levels of knockdown. Obviously in the caudate and the putamen where we actually do the injection, but also in areas of the cortex and thalamus and other places where you would want to see the knockdown, despite the fact that we didn't deliver it specifically that area. And we have begun the phase 1/2 study, and this just summarizes what we're doing in phase 1/2, including various measurements of efficacy and other elements of the clinical trial. We're planning to dose about 25 patients. We're going to use imitation surgery to have a form of placebo, if you will, or at least a control. And we'll be treating early manifest patients, so we can see early on some of the benefits of what the treatment is providing. Next, I'll just cover some of the earlier pipeline as well. So our Hemophilia A program, as I mentioned, is not a traditional factor 8 replacement, but rather a novel factor 9, which is able to be normally activated. It isn't actually activated when it's made, but it is normally activated through normal mechanisms, and then is able to go on to activate factor 10 without the need for factor 8. So it's factor 9 that's independent of factor 8. It's a very interesting product, and there's a number of advantages that I can cover with it, but it is described here what we anticipate the advantages over factor 8 replacement to be. And so this is a very interesting program that we think will be very differentiated. And this just is data that shows we're able to achieve in non-human primates levels of expression that we think will be therapeutically relevant for clinical trials. The next is our Fabry's disease program, which we use a modified NAGA rather than alpha-gal, which alpha-gal is the protein that's deficient in Fabry's disease, but NAGA is a very similar and closely related protein. We've modified it slightly so that it actually has alpha-gal activity, and it is more, we believe this will be more effective than alpha-gal for the reasons that are described here. It also will be effective in patients that developed inhibitors to alpha-gal, which is a problem in Fabry's patients. And in and of itself, even with the modifications, this is a non-immunogenic protein. So we're very pleased with that program. This is some data just to show that in the target organs, it's very critical. Several of the target organs include kidney and heart. We target the liver with the delivery, but then the NAGA protein, the modified NAGA, goes into the bloodstream, goes into the kidney and the heart. And this shows a higher level of knockdown of GB3 in those target organs. So that's a good reason for us to want to proceed with this program. And then our SCA3, which is cerebral spinal ataxia. We are also using our knockdown approach to then inject directly into the brain as well. And silence the SCA3 microRNA or messenger RNA and knock down the SCA3 protein, which is a deleterious mutated protein in these patients. And so we are leveraging our experience with the Huntington program to do that. And this is some data that indicates that we are achieving meaningful levels of knockdown in mice. And we believe we can move this program very quickly based on our Huntington experience. So we know what we're doing in this area very well. And that this just summarizes what, again, our near-term goals, which I went over before. That really is the conclusion of the presentation. And I don't know if we do questions or if we have time for questions, but I'm happy to take questions. Thank you. I have 1 minute left. Somebody have a question? Yeah.
Hi, very nice presentation. You mentioned that there is no T-cell pre-existing. Is that only for that screening, or do you check also there?
Well, once you administer the therapy, then yes, there's no T cell response whatsoever, but there's an antibody response. So you will have, yes. There's no T cell responses ever been observed, but antibodies will go up once you treat with the AAV gene therapy, against AAV5. But they don't tend to be pre-existing antibodies, and we never see a T cell response, which is very important for a number of reasons, but it's certainly for durability. Yeah. Mm-hmm.
I'm in the T cell field, so it's surprising to see that you get responses for antibodies, but there are
But no T cells. Yeah.
That was
Yeah.
the reason why I asked.
Agreed. Anything else?